All right, for our next topic, we're going to start talking about what drugs do to microbes. What's the mode of action? How are they actually killing those microbes? All right, so again, that gets into mode or mechanism of action. We've already talked about how we want our antimicrobial drugs to be selectively toxic, so be sure you understand what that means. Now, what about, at different times during the class, I have mentioned that there's a difference between prokaryotic and eukaryotic cells, and I mentioned how this was going to come back up again later during the semester, and so here we are. We have a lot more antibacterial drugs than drugs that impact other types of microbes, and that's because bacteria are prokaryotic organisms, and your fungi, your prokaryotes, and your helminths are all eukaryotic organisms. And then viruses, of course, are off in a category all on their own. All right, so fungi, protozoans, and helminths, they're eukaryotes. That means that their cells operate in a much more similar way. Their metabolisms are much more similar to those of our human cells than would be the case with bacteria. So it's a whole lot more difficult to come up with and find drugs that are selectively toxic for eukaryotic microbes. With bacteria, we have a much bigger arsenal. And then with virals, it's just been difficult to come up with antivirals that home in on something that the virus does that does not also impact something that's going on with the metabolism of our own cells. Okay, be sure you take a look. There's a big table in your textbook that has an overview of many different types of antimicrobial drugs, and they're grouped together very nicely by their mode or mechanism of action, the chemical classes that they fall into, and that's going to give you a pretty good overview. Use your review guidelines or study guide for Chapter 10, because I'm not going to require that you go through this table and memorize all these different drugs and exactly what they're doing. We're going to talk about some of these on a broader scale, so the things that we're going over here in the video lectures and or the things that I've got listed in your study guide learning objectives for the chapter, those are the things you're really going to want to home in on. Another thing I want to point out too, first generation drugs. When you hear of this drug, you might read sometime this is a first generation antibiotic drug. That means it's an early version, an unimproved version of the drug. Later generation drugs, like second, third, fourth generation, those have been improved. So if it was an antibiotic first generation, it's probably a semi-synthetic now. It's been tinkered with by the chemists to try to improve it and make it work better, or to have a broader spectrum or spectra. So it works against a broader array of microorganisms. So this is a pretty good illustration from your textbook, which is highlighting mechanisms of antimicrobial drug action. And of course, this is a bacterial cell, so most of these are going to be applying mainly to bacteria. But some kind of major categories here: protein synthesis, acting on the ribosomes. Lucky for us, bacterial ribosomes are a little different from eukaryotic ribosomes. So therefore, we've lucked out, and there are quite a few drugs that in some way interfere with bacterial ribosomes but don't interfere with the ribosomes in our cells. And of course, if your ribosomes are screwed up, you can't make your proteins. Enzymes are mostly proteins. If your enzymes aren't working, your metabolism is not going to be working and you're going to die. And you can read through some of the examples of antibiotic drugs or antimicrobial drugs that work through the ribosomes by screwing those up. Erythromycin, you've probably heard of that one before, maybe tetracyclines. There's a whole bunch of them. Cell wall inhibitors. So back in Chapter 3 when we were talking about the peptidoglycan cell walls of bacteria, I mentioned that quite a few antibacterial drugs that we take work by preventing the synthesis or the repair of the peptidoglycan cell wall of bacteria. And if bacteria don't have intact cell walls, they're very prone to taking up water through osmosis and bursting. They blow up and they die. Some drugs impact cell membranes. There aren't very many of these because it's hard to find drugs that will damage the cell membranes of microbes but not also damage our cell membranes. Polymyxin is one. There are antimicrobial drugs that inhibit DNA or RNA replication or transcription. Some of those are out there. They're a little bit fewer in numbers. There are drugs that impact other metabolic pathways, other chemical pathways inside microbes. There are quite a few antibacterial drugs disrupt folic acid synthesis in bacteria. So and those include all the sulfa drugs, the sulfonamides. We'll talk a little bit about those as well. So a little bit more about the antibiotics or antibacterials, because if we're thinking about antibiotics as naturally occurring drugs and semi-synthetics as antibiotics that have been tinkered with, you want to put those together. And probably would be better to say antibacterials here that inhibit cell wall synthesis. So this is pretty common for antibacterial drugs to disrupt the production or the repair of the peptidoglycan cell wall. Remember, bacteria have that, our cells do not. So that's good. You want to find a drug that screws up some sort of microbial structure or process that doesn't exist in our cells. So if you guys remember, peptidoglycan consists of repeating sugars named NAM and NAG. You've got these NAM-NAG sugar strips they're showing here. And then they're held together through little chains of amino acids, little peptides. Those are cross-links. So they cross-link those sugar strips this way and then also this way. So you have a 3D cell wall structure. So guess what? If you can't build those cross-links, there's nothing holding those NAM-NAG sugar strips together. So the bacteria actually produce the NAM-NAG sugar strips and they secrete them outside to help start building the cell wall, but there's nothing holding them together, so they're just going to kind of go floating off into Never Never Land, I suppose. All right, so drugs that interfere with cell wall synthesis in bacteria, most of them fall into a category called the beta-lactams. That's a class or a category of lots of antibacterial drugs, and they work. So there are enzymes that build those cross-links, and these drugs inhibit those enzymes. Remember, we talked about enzyme inhibitors back in Chapter 7. Penicillin, cephalosporin, maybe you've heard of that before. And this says penicillins and cephalosporins because there are different versions of those chemical drugs. Vancomycin, maybe you've heard of that one before. That also interferes with some types of cross-linking bridges. So these cross-linking bridges, these peptides can differ a little bit from some types of bacteria to others. Another feature, these drugs only work on growing bacteria. So if a bacterial cell has already built its cell wall and that cell wall is stable, it's not going to be killed by this particular drug. But bacteria generally need to continue reproduction in order to maintain an infection in your body. So even if the existing bacteria aren't going to be killed by the drug, any new ones that are produced as those cells eventually begin to divide will be. A little bit more on the beta-lactam. So you do need to know that there is a class of drugs called the beta-lactams. You need to know that those interfere with peptidoglycan synthesis in bacteria, and recognize that penicillin is a member of this group. Methicillin is a member of this group, since we hear quite a bit about methicillin these days as well. And you can take a look in your textbook at some of the other drug types that fall into this category. Amoxicillin, pretty commonly given for, say, ear infections in kids. That's another one of these beta-lactam type antibiotics. And some of them are natural antibiotics, and then some of them are semi-synthetic drugs. Where does this name beta-lactam come from? So this is the actual chemical structure of penicillin. This is produced by penicillium mold. And now right in here, this is one of those organic chemical structures. So you've got this square-shaped area here. So there's a carbon right there, a carbon right there, a carbon there, not drawn in. and some hydrogens that are not drawn in as well. This is called a beta-lactam ring. So all of the beta-lactam antibiotics have that as a feature of their chemical structure. So here's methicillin over here. So it looks quite a bit like penicillin, but the chemists came along and added an oxygen here and a carbon and 3 hydrogens right there, and then also down over here, and that's what makes this methicillin a semi-synthetic versus penicillin a natural antibiotic. But both of those work the same way. They have a very similar chemical structure, and they disrupt peptidoglycan synthesis. Now, methicillin, the reason we're hearing a lot about methicillin these days is maybe you've heard of MRSA before, M-R-S-A. That stands for methicillin-resistant Staphylococcus aureus. That is Staphylococcus aureus is a pathogen and can cause many different types of infections. And typically, if you had an infection with Staphylococcus aureus, you were prescribed methicillin to combat it. However, because methicillin has been so widely used, there are many strains of, there's a strain of Staphylococcus aureus now called MRSA that's resistant to methicillin. Methicillin doesn't work on it anymore. And our drug arsenal is pretty limited for that organism. One of the things that is sometimes used is vancomycin instead of methicillin. Vancomycin has some drawbacks with it. And unfortunately, though, there are now Staphylococcus aureus that are becoming resistant to vancomycin. So now we have VRSA, vancomycin-resistant Staphylococcus aureus as well. All right, how about all right, so those are antibacterial drugs that interfere with bacterial cell wall production. What about antibiotics or other antimicrobials that might inhibit protein synthesis? So here we're really, the example here in the diagram, we're taking a look at antibacterial drugs because these drugs are going to screw up the function of bacterial ribosomes, but hopefully not our ribosomes. And quite a few, take a look at the chart in your textbook, there are quite a few antibacterial drugs that fall into this category. So there are different groupings of these based on having kind of similar chemical structures, like the aminoglycosides, which include streptomycin, gentamicin, neomycin, that's present in Neosporin ointment if you've ever used that. So those are all in that category. Tetracyclines, that's another group or class of antibiotics. Macrolides, like erythromycin, azithromycin, if you've ever heard of a Z-Pak before, that's azithromycin. That's a macrolide type of antibiotic. So this diagram is showing you some of the different ways that these ribosomes get screwed up. So some of your aminoglycosides, this group, attached to the 30S small subunit of bacterial ribosomes and warps the shape. And so during translation, the wrong amino acids are incorporated into the protein that's being built. Tetracycline, some of the aminoglycosides that are out there, attach to the ribosome and they block a tRNA from bringing in the next amino acid in a protein. Chloramphenicol prevents the peptide bonding between amino acids that occurs during translation. There are some macrolides that prevent a ribosome from shifting, so it can't move from one codon to the next. So that shuts down translation as well. So a number of different ways that protein production can be disrupted by those types of antibacterial drugs. All right, so I'm going to go ahead and cut this video clip off here. In the next one, we'll continue to talk about some of these different types or classes of antimicrobial drugs and how they actually kill them, how they actually work.